Okay, guys, so we're continuing our neuron lecture. This is the second video in the series. If you have not done so, please make sure you're familiar with RMP. That stands for resting membrane potential. So make sure you're comfortable with resting membrane potential before you proceed. If not, go rewatch it again because you'll need to understand that in order to understand action potentials. Okay, so at this point, we've talked about resting membrane potentials. Remember that inside of the neuron, it is negative. Remember that you have more sodium outside of the outside of the neuron than inside, and you have more potassium inside than outside. Okay, so again, review that if needed. Now we're going to talk about excitability, which is the ability to change membrane potential so that you can change the internal charge. So before we get onto that, let's just back up and talk about what electricity is. Electricity just means movement of charges. Now, typically when you think about movement, like for example, movement of the electricity through like lights, those are movement of electrons. But for the neurons, when we create electricity, what we're moving around are ions. And those ions are going to be positive ions, specifically sodium and potassium. So we're going to create movements or flows of those positive ions, and that is going to create the electricity that we're going to use for an electrical signal. Okay, so we're going to create that change or that flow in ions by changing the permeability by opening and closing voltage-gated channels. Okay, so let's start with some vocabulary that you're going to need to know. The first one is depolarization. Depolarization. Now, keeping in mind that resting membrane potential is negative, depolarization means becoming more positive. Now, it doesn't necessarily mean it has to be all the way positive. It could still be negative, but if it's less negative, that is the same as more positive. Okay, so becoming more positive inside of the cell, that's depolarization. And repolarization means returning back to the normal negative resting membrane potential. Okay, repolarizing as in return to resting membrane potential. Now, hyperpolarization means that instead of becoming more positive, you become more negative. You're even more negative than resting membrane potential. Okay, so we're going to start with how those neurons create action potentials, but we need to start way back at the beginning of the story with a stimulus. Now, a stimulus just means that something has changed. There's some sort of physical response that causes a change in the cell. Now, if you're talking about sensory neurons, those could be changed like light, for example. If you're talking about photoreceptors in the eye, those can be changes like mechanical changes. If you're talking about like the receptors in the ears or the sensory neurons in the skin, any of those could act as a physical stimulus that could cause a graded potential in a neuron. Or it could be a chemical, such as the taste receptors. They respond to specific chemicals. Or it could be the neurotransmitter from another neuron, like what happens when all the neurons in the brain are talking to each other. Okay, so you have some sort of physical or chemical stimulus that creates a graded potential. Okay, so step 2, we're going to create a graded potential. Now let's look at this word graded. So if you remember back to the muscle lecture, what did graded mean in the muscle lecture? It meant that the amount of force varies. So again, graded means varies in physiology. Okay, so if we're talking about a graded potential, and potential is basically the internal charge, we're talking about varying the internal charge. Those are graded potentials. Now, what we want to really focus on is the difference between graded potentials and action potentials. We're going to compare them at the end of today's little mini lecture. So, when you have a kind of stimulus, for example, if you are a nerve ending in the dermis of the skin, you can have just simple touching, something like that. That's going to trigger a small localized change in charge. Now, that small localized change in charge can either be towards positive, okay, so something that creates a depolarization, or it could be making it even more negative. That would be hyperpolarization. Okay, so you have a stimulus that can cause either graded depolarization or hyperpolarization. And because these are graded potentials, these are going to be small localized changes in charge, localized changes in charge, and they will travel outwards. They will affect and spread out from the central point of the stimulus, but because they are graded potentials, they are going to decay the further away they get from the original point of contact. Right? So if I touched it right here, you can create a depolarization wave, but the further away we get from the original point, the depolarization will be less and less as it goes outwards. It kind of falls away as it goes outwards. But that will be different when we talk about action potentials, which can be conducted. So we'll talk about that later. Okay, so at this point, we create a small localized change in charge, either more positive depolarization or more negative hyperpolarization, and which one it is entirely depends on what type of stimulus it is and what type of receptor it is. For example, in the eye, a lot of those receptors respond to hyperpolarization versus depolarization. But so whichever it is entirely dependent upon what type of stimulus and what type of receptor it is. Okay, so for now you just need to know depolarization more positive, hyperpolarization more negative. Now, those graded potentials also allow for summation. Okay, so summation for muscle physiology, remember that summation means when you increase the stimulus of increase the frequency of the stimulus, you increase the force. Okay, so again, you get a varying amount of force. And what summation means for neurons is that they can add on top of each other. So the depolarizations will add on top of each other. Or if it's a hyperpolarization, they can subtract from each other. Okay, so let me go through it real quick. So in the orange here, here's our neuron. And this neuron has a bunch of dendrites, and those dendrites will synapse or connect to the axon terminals of another neuron. Okay, in this case, we've got one single neuron with two other neurons talking to it. Now, if one of the neurons talking to it creates a depolarization, and the other one creates a hyperpolarization, because you have one increasing the you have one depolarized making it more positive and one hyperpolarized making it more negative, they end up canceling each other out, and that's the end of the story. It just goes back to resting membrane potential. But if both of the neurons that are talking to it sum on top of each other, both of them add depolarization, so positive, this one will stack on top of the other positive, and that'll create a double amount of increase in the charge, right? So it's even more positive. And looking ahead, if it's enough, that can reach what's called the threshold, and that will trigger an action potential, which is shown in yellow here. Okay, so for now, just think about depolarizations summing or adding up to each other to become more and more positive and getting closer and closer to a threshold that can trigger an action potential. Okay, so that can either be two ones talking to it or one single neuron sending a whole lot of stimulus, and a whole lot of stimulus results in summation up towards the threshold. Okay, so depolarization, sum on top of each other, hyperpolarization, subtract. And that's why they're graded. They're graded because they can do summation, and therefore they can vary the strength of the change in potential. Okay, so once you have one spot on the neuron creating those graded potentials will spread, and as they spread, they will decay. So what happens is usually the stimulus will occur on one of these dendrites, and then it will spread in all different directions. And as it spreads, it's going to spread outwards towards the cell body, and then eventually, if it's a strong enough depolarization, or if it's multiple depolarizations that have some on top of each other, eventually it'll reach the axon hillock, which, as you recall, is just the very initial part of the axon. Okay, so once it reaches the axon hillock, now we're going to take those graded depolarizations that have stacked on top of each other, and if they are strong enough to reach the threshold, then we're going to change those graded depolarizations into action potentials. Okay, so now we're going to talk about, well, what is an action potential? How is it different from a graded potential? But before we do that, let's ask a question. Well, why? Okay. Oh, wait, I'm sorry. I forgot. Let's talk about what an action potential is first. So action potential. So remember, graded potentials means it varies in charge. Action potentials are all or nothing. And so what that means is they only occur if the potential, how positive it is, reaches a very specific threshold. Once it reaches the threshold, the depolarization phase, how positive it becomes, acts the same way every single time. So it does not do summation. As once you reach the threshold, the strength of the stimulus no longer matters. If you increase the strength of the stimulus, it's still going to do exactly the same pattern. It'll go and have a depolarization phase followed by a repolarization phase. Okay, so remember that for graded potentials, you either have depolarization or you have hyperpolarization. When I say action potential, I mean depolarization followed by repolarization, always both pieces. Depolarization plus the repolarization creates one action potential. So everything you see here, this is one action potential. Okay, so again, all or nothing, no summation can happen for the action potentials. Okay, so now let's go to the question I thought we were going to do is, well, what's so special about the axon hillock? Okay, so if you recall, up until this point, once you have a stimulus, you just have graded potentials sort of spreading and decaying outwards, and you don't create action potentials until you reach the axon hillock, which is the very beginning part of the neuron. Okay, so what does the axon hillock have that the other places do not? It has a specific structure, and those are voltage-gated ion channels. Okay, so remember in the previous lecture, we talked about the 3 different types of ion channels. You have leakage channels that are always open at all the time. You have the sodium-potassium pump, which is again always pumping all of the time. Then you have the voltage-gated ion channels. And the voltage-gated ion channels, remember, they are voltage-gated because they only open at a particular charge and then close at a particular charge. They are only physically present in the axon. So you can only create action potentials from the axon hillock along the axon, not along the dendrites or the cell body. Okay, so let's look at those voltage-gated channels. Okay, so voltage-gated, what did that mean? It means they open at a particular charge and then close at a particular charge. Okay, they have a range of charges that allow them to be open. And at rest, so during resting membrane potential, the voltage-gated channels are going to be closed. Okay, so we have voltage-gated sodium channels, Na stands for sodium, and voltage-gated potassium channels, K stands for potassium. At rest, both of them are closed. Okay, so nothing's getting through those right now. Now, if you have enough depolarizations, so you have graded depolarizations that stack on top of each other, so if they're enough to reach that threshold, what that's going to do is you now reach a positive charge that's enough to open those voltage-gated sodium channels. Okay, when the voltage-gated sodium channels open, what's going to happen to the sodium? Okay, so recall back to the resting membrane potential. For the resting membrane potential, is the sodium inside the cell or outside of the cell? Sodium is outside of the cell. So now what we have is a bunch of sodium outside of the cell waiting to get through, right? Very steep concentration gradient for that sodium. So when the voltage-gated sodium channels open, what's going to happen to the sodium? The sodium is going to flood into the cell, right? So the resting membrane potential created those steep concentration gradients that allowed the action potentials to work, right? They set the stage for them. The sodium will flood into the cell. Okay, now we have a bunch of positive ions coming in. When you add a positive, what happens to the internal charge? It becomes more positive. Okay, so add a positive, you become more positive. And what happens here is, you see how you have this rapid jump up in the charge inside of the cell. It becomes very positive very quickly. That's because the more positive it becomes, the more of those voltage-gated sodium channels are going to open, the more sodium will flood in, more. What does that sound like? The opposite of a negative feedback loop, it's a positive feedback. Okay, so that's why you have this rapid increase in charge. It gets very positive very quickly because you're adding a bunch of positive sodium into the cell, a whole bunch of positive sodium into the cell makes it very positive very quickly. Now, what happens here? Okay, at this point, we have a positive feedback loop, but it's not going to go on forever. What happens here at the peak? Now remember, when I say voltage-gated channels, open at a particular charge and then close at a particular charge. Okay, so remember, it's a range. Once it becomes too positive, those sodium channels are going to become inactivated. And when they're inactivated, that means that the sodium can no longer come in. So no positive ions coming in at this point. The second thing that happens at this time is that the voltage-gated potassium channels, which were closed, now open. Now, thinking back to the resting membrane potential, what happens to the potassium? Potassium was hoarded in the cell, so you have a lot of potassium just held up inside of the cell. What happens to that potassium when you open the gates? The potassium is going to flood out. Okay, so the potassium will flood out of the cell, and when it floods out of the cell, when you are losing a positive, you become more negative. Loss of a positive makes you more negative. So the inside of the cell will become much more negative. See how it drops down here? It's becoming more negative, and it's going to return towards resting membrane potential. That's called repolarization. Okay, so let's give you a summary here to hold on to. So here at this point, we have resting membrane potential. Notice that resting membrane potential is negative, usually around -70, but don't memorize that number. So here we have negative resting membrane potential. You're going to have some sort of stimulus that creates a localized depolarization. If the depolarization is strong enough or summed up enough to get to the threshold that'll cause the voltage-gated sodium channels to open. When the sodium channels open, because you've already used the sodium-potassium pump, the sodium is going to flood into the cell. When you add a positive, it becomes more positive. So we have an increase in charge. At this point here, the sodium channels will then close again because it's too positive now. They'll become inactivated. And at this point, the voltage-gated potassium channels are now going to open up. Okay, they're like, oh, nice, I want to open up now. That causes the potassium then to flood out, okay, because of the resting membrane potential that we've already created with a sodium-potassium pump. The potassium floods out, and when you lose a positive, you become more negative. Okay, so we have a decrease in charge. See how it drops? And that's called repolarization because you're returning back towards your resting membrane potential. So for an action potential, two phases: depolarization followed by repolarization. This whole thing, one single action potential. Okay, now we need to talk about this part that I keep skating over. Okay, so here we have our depolarization becoming positive, our repolarization returning back to negative. Okay, but at this point right here, we have what's called an undershoot or refractory period. So let's talk about refractory periods. There are two refractory periods. Refractory periods means that the neuron is unable to create an action potential during that time. So there are two parts. There are two different refractory periods. The first one is called the absolute refractory period. Absolute as in absolutely no new action potentials during this time. And this is why action potentials cannot do summation. They cannot do summation because you cannot create another action potential when you're already doing one action potential. It is entirely impossible. And the reason for that is either the sodium channels are open and the sodium's flooding in, so you're unable to reset up, or at this point here, remember that I said that the voltage-gated sodium channels are inactivated. What I mean by that is they're not just closed. They're not just closed. Instead, what it is see this little yellow ball right here? They are inactivated. That means that this little yellow ball right here swings up and literally blocks the voltage-gated channels. Okay, so they're not just closed, they're full-on blocked during the repolarization part of the action potential. So because they're either open and it won't work because the sodium's already flooding in, or they're full-on blocked and therefore you can't create another action potential at that time either. Okay, so because they're blocked during that period, that makes it so that you are unable to get any kind of second action potential while you're already doing one action potential. Because they can't happen at the same time, that's why you're unable to do summation. So action potentials cannot do summation of any kind. Now, there's a second refractory period that happens after the repolarization. This undershoot part right here, this is called the relative refractory period. So relative means that it is possible to create another action potential at this time, but it is very difficult to do so. And the reason for that is because during this period right here, the voltage-gated potassium channels are open. That means that that potassium is flooding out of the cell. So you're losing a positive. That makes it so that it's very negative. If you're losing a positive, it's going to make it very, very difficult for you to return back up to the threshold. Look how far away you are from the threshold. So you would have to have an extremely strong stimulus or a whole bunch of stimulus, some kind of creating graded potentials that sum on top of each other in order to get up to the threshold in order to create another action potential. So relative means that it is possible to create one, but it would have to be a very strong stimulus to provide enough positive membrane potential to return back up to the necessary threshold. Okay, so absolute, cannot do it, doesn't matter how the strength is. Relative, can do it, but it have to be very, very strong because you're already so negative. You have to get all the way back up to here. So that is why action potentials are all or nothing. They cannot do summation because of those refractory periods. And so that means that even if you have an increase in the strength, you cannot create a stronger action potential. Okay, once you reach the threshold, it fires the same way every time. Okay, so if you look at the amplitude here, the change in voltage is exactly the same. Whereas graded potentials, they're going to vary in how the change in voltage and the amplitude of the chart. Okay, so even with the strongest one, they look the same as this one. So if you can't change the strength of the action potential, how does your brain know how strong the stimulus is, right? So how do you know if you have just a light touch or a dark or deeper pressure? How is your brain going to be able to determine the strength of the stimulus? You need some other way to encode that information. So it's not the amplitude of the action potential. What it is the frequency of the action potential. Okay, so if you see that every single action potential is exactly the same way, but as you increase the frequency, increase the force or increase the strength of the stimulus, you're going to increase the frequency of the number of action potentials that are created. And that's because you're just going to reach that threshold sooner. And once you reach the threshold sooner, you're going to create an action potential. So your brain is able to interpret this increase in frequency as a stronger stimulus strength. We'll talk about that again when we talk about the sensory neurons in next lecture. So what I want you to do right now is pause the video. I want you to try and create this chart right here and try to fill it out. And then once you are done with that, come back and we'll go over it together. Okay, welcome back. Okay, so for the graded potentials, okay, the graded potentials will be found in the dendrites in the cell body. Action potentials cannot be created until you reach the axon hillock or in the axon. That's because you don't have the voltage gated channels that you need in order to create action potentials until that region. And then, so yeah, you don't have these voltage gated channels. So if you don't have the voltage gated channels, then you can't create those action potentials. Now remember that graded means varies, which means that you can change the amount of charge, and changing the amount of charge is done through summation. So remember that depolarization increases it, so they add on top of each other, whereas hyperpolarization decreases it and it subtracts it back down. So they do have the ability to cancel them out if you have both depolarization and hyperpolarization at the same time. Okay, and the reason you don't have summation is because of those absolute and relative refractory periods that make it very difficult to have an action potential occurring at the same time. And if they can't occur at the same time, that means that they can't stack on top of each other. So no summation. Okay, that's enough for now. Go on to the next video. Stop recording.